Good afternoon, everyone. This is Niharika Chaudhary. I'm a research scholar at Department of Physics and Computer Science, Dayalbagh Educational Institute, Agra, India. First of all, I would like to thank the CSIT conference for giving me this opportunity to present my work on this platform. So thank you very much. Today's my topic of presentation is progress on protein structure prediction using various soft computing techniques. So these are my contents that I put in. Okay, let's begin with the introduction part. So basically, proteins play a vital role in all living organisms. Proteins are basically the complex macromolecules that is composed of 20 amino acids that has been arranged in a linear chain via peptide bonds. These amino acids are the building blocks of all natural proteins that can be classified into hydrophobic and polar. Hydrophobic, we can say, that mixes with water, and polar, that doesn't mixes with water, it's hydrophobic, and polar, that mixes with water. So in that way, we have been classified into two. So the prediction of 3D structure of protein from its primary sequence, or we can say a linear chain, is established a PSP problem. PSP is one of the open research area in structural bioinformatics, right? Many experimental techniques like NMR, that is nuclear magnetic resonance spectroscopy, and X-ray crystallography has been widely used to determine the native structure of proteins, but due to the growth size of protein data bank, these methods actually fails. And also these methods are not always reliable, are very cost effective, and are not that effective. That's why these above methods fails. Okay. Now what is protein structure prediction? So this prediction problem is predicting the 3D structure of protein from its primary sequence is a PSP problem. So understanding the 3D structure of a protein from its one-dimensional amino acid sequence is itself an unsolved problem, because only the 3D structure of any protein determines the biological functioning of any protein. So the tertiary structure is the global three-dimensional structures. Here, biologists describes a hierarchy of protein structures with 4 levels that has been explained in the next slide. Okay, different levels of protein structures. The first structure is called the primary protein structure. Here, we have 22 amino acids in existence. So 22 or 20 amino acids are interlinked together and forming a polypeptide chain, which is the sequence of amino acid, and we can say it's a primary protein structures. Every protein structure undergoes a process of folding and then converted into beta sheet and alpha helix. When it gets converted into beta sheets and alpha helix, the secondary protein structures comes into existence. Then secondary protein structure itself goes into a process of folding. Here I'm using a word protein folding. Protein folding actually happens due to the hydrogen bonding of bonds, due to the high temperatures or like acid basic interactions. Due to these hydrogen or high due to these components, it goes into a process of folding. So further secondary structures folds, and then it's get converted into tertiary protein structures. Then tertiary structures again goes into process of folding, and it gets converted into quaternary protein structures, which is a combination of multiple polypeptide chains. Okay, what is the relevance of PSP under my study? Actually, PSP is an NP-hard problem. It belongs to an NP-hard problem, and the factors that makes the PSP a tough problem are: first, there is a structure sequence gap. What is structure sequence gap? It's a gap between the number of sequences and the known structures. So the problem is it's very huge. So we have to reduce this structure sequence gap here. As we can see in UniProt KB database, protein number of protein sequences that it contains are this much: 21, 91, 74, 961. And whereas the number of known structures, protein structures that has been stored in protein data bank are this much. So we can see how much gap is there between the sequences and the structures, known structures. So it's a problem. So we have to reduce this structure sequence gap. That's why PSP is relevant. So the next problem is problem of protein folding. During the synthesis, the protein folds into a unique three-dimensional structure, which is known as protein folding process. So we have to solve this particular problem also, because if any protein, if folds into an incorrect manner, then it can lead to misfolding of any protein, and a number of diseases like Alzheimer, cancer, etc. can be occurred, which is not good for any human organism or any organism. So that's why we have to make sure that protein should not misfold. That's why problem of protein folding is also very relevant in PSP. Next one, finding the most stable conformation of protein in a huge search space. We all know there is a huge search space stored in many databases of protein structures. So we just have to find the most stable conformation, that is the most native structure of protein we have to find. And the most native structure of protein will be whose potential energy function will be minimum. So here we have to find the global optimization of any protein energy function. Okay, related work on PSP using soft computing, as it's just a survey paper. So whatever the work that has been done on protein structure predictions using soft computing techniques, I have covered these techniques and it has been put up in here in this presentation. So basically these 6 techniques belongs to soft computing techniques. So let's get started with the first one, computational approaches. Okay, computational approaches basically categorized into two. First is template based modeling and free modeling. Okay, template based modeling is a technique or method which makes use of a template to predict the 3D models. The protein with the same sequences are tend to bend. Okay, like homologous sequences are there. The proteins with the same sequences means the proteins which who have the same sequences will tend to bend in the same structures. That belongs to the template based modeling. Next is free modeling, ab initio modeling or de novo modeling. We can be used interchangeably. Means we can either say free modeling, we can say ab initio modeling or de novo modeling, all are same. So this techniques attempts to build a 3D structure without using templates or without using any homologous or same protein sequences. So authors here in related work have actually developed some of the tools based on TBM approaches, which I have mentioned here, like Swiss model, HH Blitz, Modeler, iTesser, Quark, Bhagirath. All are based on these template based modeling. Okay, let me take first one, Swiss model. Swiss model basically was developed by Torsten Schwede's structural bioinformatics group. It's an online tool for homology modeling of protein structures, right? The second one, HH Blitz. It's also based on TBM, which is employed for template recognition and target template alignments. Modeler tool uses the target template alignments to implement structural modeling by satisfying some of the special restraints. iTesser is a comprehensive structure prediction methods based on TBM approaches. And then Quark comes, which employs the replica exchange Monte Carlo simulations guided by the knowledge based force fields. And Bhagirath is a web server actually, which is one such ab initio homology hybrid method, which can be accessed in the form of web server. Here, the David Baker lab created a model Rosetta, which is a template based approach. And its greatest success was recorded in CASP-11, when it actually predicted the structure of sequence of 256 amino acid. Here I have used the word CASP-11. Okay, let me tell you what is CASP. Okay, I'll explain it later. Let me come to this second approach, deep learning based approaches. Basically, this CASP method is mostly used in deep learning based approaches. So, deep learning is a subfield of machine learning, which is based on artificial neural network. Actually, it used to learn the patterns from its input data and make accurate predictions in turn. And it has proven extremely effective challenging problems in tackling challenging problems of protein structure prediction when it is used in CASP-13. So this gives an encouragement to move it further to solve the problems of PSP. So CASP is known as a critical assessment of protein structure prediction. It actually assesses the protein structures predictions, whether it be a 1D, 2D, or 3D. It basically depends on the assessment, critical assessment, which is a community-wide, worldwide project that takes place biannually since 1994. So it takes every second year. So it takes place biannually. It aims the protein structure prediction methods and stimulate the advancement of the field, and its core principle depends on the fully blinded testing of structural prediction algorithms. It fully blinded tested the structural prediction algorithms in all of their like series CASP 1, CASP 2 till CASP 14. Okay, the first success in protein tertiary structure was observed in CASP 4, where it used only small proteins, that is less than 120 residues. But later, better contact prediction approaches were introduced in CASP-11, competing pipelines with promising improvements in prediction accuracies. Okay. These are some of the methods that authors have designed based on the deep learning methods. This method, deep meta PSI cove DMP, that was comes into existence in 2019, and this method mapped in 2020. So are based on deep learning, and it predicts the contact prediction. It's a contact prediction tool which combines the input features sets utilized by these two methods, MAT PSI cove and deep cove. Both these methods are the components of MapPred itself. Al Qureshi is an author. In 2019, he and other authors, his colleagues, I can say, builds the RGN, the recurrent geometric networks, which is end-to-end differential protein structure predictor. The next method is multicom, which comes in 2021. TR Rosetta comes in 2019, which is transform restrained Rosetta. It was developed by Yang and David Baker's group. I have put all these methods in references. So in paper, in review paper, it can be seen. So by Young and David Baker's group in order to predict the inter-residue geometry and orientations. Next method is Raptor X contact, which comes in 2017. It's a residual neural network based model used to predict protein contacts. Triplet rest method comes in 2021, developed by Zhang's group, is also a contact map predictor that ranked first in the contact prediction category of CASP-13 for PSP. So here we can see the CASP-13 has listed this method developed by Zhang's group in 2021 itself. DeepMind's method AlphaFold is a topmost method, I can say, that has listed in CASP 13 and CASP 14 itself. It's a very it's, I can say, it's a best tool for protein structure prediction method developed by DeepMind. And every author, or I can say daily every year researchers are using this particular technique DeepMind. On the basis of AlphaFold they are using, and they are predicting the protein structure prediction problems. So I guess it has achieved the best performance in the template based modeling domain of CASP 13, and has evolved to AlphaFold 2 at CASP 14. CASP 13 comes in 2020, I guess last year in December, 2020 December. Okay. The third technique is support vector machines. Support vector machine is also a machine learning method which is used to predict protein structures from its primary sequence. Its fundamental approach entails transforming the sample into a high dimensional Hilbert space and searching for a separation plane there. This is the related work done, which authors have proposed, like Wang et al. In 2016. I have used et al. Because there are other authors also. So these authors in 2016 proposed a protein secondary structure prediction method. Here, protein secondary structures, not tertiary and not primary, it's secondary structure prediction method, which is based on SVM with PSSM profile, that is protein specific scoring matrices. This author in 2016 introduced two unique methods like ProQ-ROS-FA and ProQ-ROS-SEN, that is modeled after cutting edge of ProQ2 technique. ProQ2 technique basically comes in 2012, which is a model quality evaluation algorithm which uses SVM to forecast both the local and overall quality of protein models. SVM QA is one of the best SVM based single model quality assessment methods, and ProQ3, which inherits ProQ2, and with additional full form Rosetta energy and coarse grained centroid Rosetta energy, hence achieve the highest average GDTS score. GDTS score basically are used to define the define the particular model's efficiencies. Neural network techniques. Neural network techniques are basically computational models which consist of small units interconnected with each other through some correlated weights, such as neurons within the brain or the artificial neural network. Here I have listed some of the authors and their related work, like Heffer et al. In 2015 and 2017, because Spider 2 tool comes in 2015 and Spider 3 model comes in 2017. Both are based on deep artificial neural network, and Spider 3 is based on long short-term memory LSTM and bidirectional recurrent neural network BRNNs for protein secondary structure prediction. Spencer et al. Proposed the first deep learning based PSSP method called DNSS. JPRED and Porter 5 are the tools that are the most accurate predictors, which is used in protein 3 class secondary structures. Scorpion, that comes in 2014, predicted the 3 common secondary structure states, whereas this method mufold SS in 2018 transfers the input matrix to either 8 or 3 secondary structure states. Basically, we have states of secondary structures. Either it can be 3 state or either it can be a 8 state, or we can also say Q3 and Q8. Okay, fifth one is evolutionary computational techniques. Evolutionary algorithms and genetic algorithms. These are the two subtypes of evolutionary computation. Different protein structural representations such as dihedral or torsion angles and lattice models. These are the calculation part of protein structures which may be used in EC based methods. So here is the related work done by the authors. In 2019, Biomedine et al. Used evolutionary programming by applying a hybrid algorithm combining genetic algorithm like taboo search strategy and local search procedure to address the 2D HB triangular model of protein structures. Don et al. In 2013 proposed a genetic algorithm which is based on knowledge, aiming to reduce the size of conformational search space. A hybrid genetic algorithm and taboo search algorithms were used by Borghese and B et al. In 2017 for preventing premature convergence and providing a faster convergence rate in solving PSP problem. Here in 2017, a mimetic algorithm based on fragment assembly techniques identified as a search heuristic of Rosetta ab initio protocol. And in 2017, authors attempted a multi-objective evolutionary algorithms for protein structure predictions, where they use 66 benchmark proteins and used to verify the for verifying the proposed method, they use the 66 benchmark proteins. Swarm intelligence techniques. Basically, swarm intelligence technique is a sub area of nature inspired computing techniques. Nature inspired computing is an emerging technology which is inspired by nature's behavior in numerous situations and can be used to solve complex problems. Swarm intelligence also based on same, as it's a sub area of NIC. Uh, it's a study of cooperating behavior of simple individuals like ants, bird flockings, bees, and so on. So, in diverse centralized systems, it's a study of simple individuals' behavior. So, there exist some techniques to develop powerful solutions to optimization problems, uh, because where optimization problems comes, we generally use nature inspired techniques because they are very efficient and they have immense power to solve the problems, complex problems or NP hard problems. Some of the algorithms of swarm intelligence are colony optimization, ACO, PSO, particle swarm optimization, B's algorithm, we can say BCO, intelligent water drops, and etc. So the related work done on the swarm intelligence technique is listed below. Like in 2010, authors created a quantum behaved PSO like QPSO, and improved PSO for protein folding prediction was done by Zhao et al. In 2014. Then enhanced and artificial bee colony ABC and its variants of ABC algorithm for 3D PSPs presented by this particular author in 2015. And in 2020, a unique multi-objective particle swarm optimization, that is multi-objective PSO, is used for a structure refinement protocol. And then in 2017, self-organizing maps, which is based on PSO approach with the efficient classification of secondary and tertiary, was explored by this particular author in 2017. Okay, these were the basic techniques of swarm intelligence and their related work that have been discussed here. So it's a summary. It's a summary of protein structure prediction tools. These all are the protein structure prediction tools. Here I have summarized that the name of method or tool, its data size, its architecture, that means from which class it belongs to, the year of existence, and the references. So these are the basic tools like ProQ2, Porter, Bhagirath, Scorpion, Spider, Rosetta, and I have explained all these tools in earlier slides. So these are the summarized form, and these are more methods or tools which I have listed here. That's it. Thank you so much. Any questions?